Good afternoon. My name is Majed. I'm a Year 2 medical student attending here at the University of Sharjah. Today I'll be explaining the urea cycle along some abnormalities that accompany it. So we have the amino acid structure, which is an important thing to understand this lecture. We have a alpha carbon group, we have a side chain, and we have a carboxylic acid group, and we have an amino group. Okay. This amino group is very important as it serves to protect these amino acids from breakdown or usage. So we cannot unleash the capabilities of these amino acids unless these amino groups are removed. Okay, and another thing to understand is amino acids cannot be stored. Unlike other structures such as glucose, which can become glycogen and stored in that form, or fatty acids that become into triglycerides and into adipose tissues, this does not happen. This is not the story here. So these amino groups serve as protection from oxidative breakdown, and our goal here is to remove these amino groups, in amino acids, and incorporate them where? Into the urea cycle. We don't like leaving free ammonia. It's highly toxic. If we do that, we would have an abnormality, which is hyperammonemia. So we want to incorporate this free ammonia finally into the urea cycle, which we're going to discuss. Urea is a very soluble material that can be excreted in urine easily, and it's not toxic as compared to ammonia, which is 1,000 times toxic. Still very toxic. So to solve the issues and relieve that ammonia or release it, we have three important amino acids to know. We have alanine, which becomes pyruvate in the transamination reaction, and we have aspartate, which becomes oxaloacetate in also transamination reaction. This is by the enzyme AST, and this is by ALT. Please know that many of these alpha keto acids are glycolysis intermediates, such as pyruvate, or TCA cycle intermediates, such as oxaloacetate. And I have a very important point to make. We have transamination reactions for alanine and aspartate, but glutamate itself can be deamidated directly, and we can release free ammonia from it through glutamate dehydrogenase. It's one of the only amino acids that can undergo direct deamidation. But the others that I've discussed, alanine and aspartate, need these transamination reactions where alanine with alpha ketoglutarate is converted into pyruvate, which is the alpha keto acid of alanine and glutamate. So we want to generate this structure, glutamate, which is highlighted here. So that's our goal. We all want to what? Shunt everything into becoming glutamate. Same thing here. Alpha ketoglutarate, aspartate through AST becomes oxaloacetate, which is the alpha keto acid of aspartate, and we have glutamate. So the goal is to regenerate glutamate for direct deamidation. So this is a clear idea of the whole story. So if we have glutamate, no problems. We can directly deamidate it and problem is solved. Alanine and aspartate need to undergo transamination reaction, in summary, and then become glutamate, and glutamate can go or undergo direct deamidation. So now we're going to move on to transport mechanisms. So now that we have generated free ammonia from peripheral tissues such as muscles, such as other tissues, we can shunt this free ammonia and incorporate it into the urea cycle and finish things off. So the first factor to discuss is synthesis of glutamine. So we have glutamate formed in these transamination reactions and many of these reactions, and when we incorporate ammonia with it, we can form glutamine right here. As you know, glutamine formed in these tissues, it's a soluble material that can be easily transferred where this glutamine goes to the liver. Once it goes to the liver, it goes back to its original form that we want. We want glutamate to go back to its state for direct deamidation. So we have glutaminase for this process. So this whole thing is to make glutamine, which is soluble, can go and cross across the other tissues and go where finally into the liver. So urea cycle happens in the liver. This is the important point I want to make. And then we have the alanine glucose cycle. Okay, so we have glucose right here in the muscle. We form pyruvate, through glycolysis. This pyruvate and glutamate right here, we want we can have a transamination reaction by the enzyme ALT. We form alpha ketoglutarate and alanine. This alanine then crosses and goes where? To the liver. Again, this is to form, alanine forms pyruvate. Pyruvate undergoes, reactions such as gluconeogenesis and whatnot, and becomes glucose. So we form glucose right here, and this glucose comes back into the muscle and gets reused. That's why it's called a cycle. And we form what? Glutamate. So our goal right here in all of these transporting ammonia from peripheral tissues is generation of glutamate, and this glutamate then undergoes deamidation, in the liver. This is through an important enzyme which is glutamate dehydrogenase. So glutamate becomes alpha ketoglutarate, and we have free ammonia right here, which is one of the ammonias that's incorporated in the urea cycle. This is through glutamate dehydrogenase. Please note that if we reverse the step, we should use NADPH and NADP plus as cofactors. So this is a very important point to make note of. Now I'll be moving to the most important part of the lecture, which is the urea cycle itself. So we start the urea cycle with carbon dioxide, okay, and water, which becomes, bicarb. And we have this free ammonia, which is from right here, which is from where? From glutamate. And then we start this process and this first reaction by carbamoyl phosphate synthetase 1, and we form carbamoyl phosphate, and this requires ATP since it has synthetase. Remember, synthetase equals ATP. So CPS1, and we have an allostric activator, which is very important to denote. N-acetyl glutamate is important because it reflects high protein levels. So a person with high protein diet would have high levels of urea. Why is that? Because in part we have high N-acetyl glutamate, which activates allostrically carbamoyl phosphate synthetase 1. After carbamoyl phosphate is formed, and this is a very important reaction as it's rate limiting, and it limits the urea cycle. This carbamoyl phosphate then joins ornithine right here through the enzyme ornithine transcarbamylase, and it becomes citrulline. Note, these two reactions happen where? Into the mitochondria. The citrulline then joins aspartate. Aspartate, as we said, through transamination reactions, which I'll discuss right now and whatnot, we get another free ammonia. So free ammonia from glutamate. Second one is from aspartate right here, you can see. So, and then we have aspartate with citrulline, and this is in the cytoplasm. Through the enzyme arginine 6-8 synthetase, we can form arginine 6-8. Arginine 6-8 then becomes arginine and fumarate, and this is through arginine 6-8 lyase. Now, a very important point to make, which is fumarate becomes malate, and then malate becomes oxaloacetate, and then that oxaloacetate right here, okay, as we said, this can undergo transamination reaction through AST, okay? This can reform aspartate and the cycle keeps going. These are all intermediates of the TCA cycle. So it's weird, isn't the TCA cycle supposed to occur in this mitochondria? We have the same isozymes in the cytoplasm, so same steps can occur outside. We have fumarase, we have malate dehydrogenase. So things can happen outside and the cycle keeps repeating. And we have arginase at the end, arginine. We have arginase at the end right here, and we can generate urea, and this urea can be excreted in the kidneys. to the kidneys and excreted in the urine. We have ornithine right here, so we go back to ornithine. So a very important thing, which is we started with ornithine initially and we ended with ornithine. Okay, so we have the same starting and ending product, and we have an antiporter to mention. Just to mention, we have citrulline, ornithine, antiporter. So ornithine enters, citrulline exits into the cytoplasm. Very also important thing to make note of, which is distinguishing the difference between carbamoyl phosphate synthetase 1 and synthetase 2. Carbamoyl phosphate right here is an intermediate in both pyrimidine pathway and the urea cycle. So in cases of CPS2, it's responsible for carbamoyl phosphate synthesis, where in the cytoplasm, not in the mitochondria as compared to CPS1, and this is in pyrimidine synthesis. Okay, so we form carbamoyl phosphate. This carbamoyl phosphate right here then can go and continue and go to the pyrimidine synthesis pathways and some abnormalities I'm going to discuss right now. So they both share common characteristics. Okay, so it's an intermediate in the pyrimidine synthesis pathway. So the types of hyperammonemia, we have type 2, which is X-linked, and we have type 1, which is not X-linked. Okay, we do not have the enzyme CPS1, so it's not present here. So you have high ammonia levels. Simple as that. It's autosomal recessive in inheritance, so it's autosomal recessive, as the other types of hyperammonemia. But I'm discussing the most important ones right here. This type 2, hyperammonemia is X-linked. So we have a deficiency in ornithine transcarbamylase, enzyme. And when we have a deficiency in that, we have accumulation of carbamoyl phosphate. Okay. When we have a deficiency in this enzyme and accumulation of this intermediate product, as I've discussed, it goes where? It goes in chunks where into the pyrimidine synthesis pathway, and then we can form orotic acid, okay? And at the end, we form pyrimidines. But please denote that this is a type 2 hyperammonemia because it has high ammonia levels, and we have the constitutional symptoms such as tremor, cerebral edema, encephalopathy, somnolence, and at the end we have the coma and death. This is different from the pyrimidine synthesis pathway abnormality, which is orotic acidurea, although both have high orotic acid, but orotic acidurea does not have high ammonia levels and does not have these constitutional symptoms of hyperammonemia, just to make note of. Moving on to the hyperammonemia outcomes, I've discussed the symptoms right now, and cerebral edema is also caused in part by glutamine, which I'll discuss right now. So we have high ammonia levels. What we want to do is shunt or remove these ammonia levels, or just keep them down. So what we do is we have alpha ketoglutarate, and we can reverse the reaction of glutamate dehydrogenase and form glutamate right here. Okay. So these two together form glutamate through this enzyme. What this forms is a deficiency in alpha ketoglutarate, which is a TCA cycle intermediate, which is important for energy production. So now we have no energy, diminished energy production. And the other latter pathway, which is we have high ammonia levels here, and we have glutamate too. But what we do right now is, through glutamine synthetase, we create or form glutamine. What this does is it diminishes glutamate levels. This becomes low. So when this becomes low, we have diminished GABA levels. So GABA is not present because glutamate is not present. So now we have neurotransmitter imbalance, and we have high amounts of glutamine, which can cause cerebral edema and hyperammonemic encephalopathy. All in all, these are the important factors, and I've discussed the genetic causes. We also have some acquired causes, such as aflatoxins, such as portosystemic shunts, which can also cause hyperammonemia. Hyperammonemia can be, and high urea levels can be pre-renal, intrarenal, post-renal. And at the end, I'd like to thank you all, and if you have any questions, you can leave them in the comments below. Thank you.